Hello students, today's video is on about cell structure. If we study a cell under a microscope, we would come across three features in almost every cell, that is a plasma membrane, nucleus and cytoplasm. All activities inside the cell and interactions of cell with its environment are possible due to these features. Structures that are present in cell are plasma membrane, nucleus, cytoplasm, and this cytosol cell organelles and cell organelles like endoplasmic reticulum, Golgi body, lysosomes, vacuole, mitochondria. Um, Plasma membrane um, is a thin, pliable plastic outermost structure which envelops the cell. It consists of a bilipid layer with embedded proteins that are integral proteins, peripheral protein. Nucleus. Nucleus has a double layered covering called nuclear membrane. Nuclear membrane has pores of diameter about 80 till 100 nanometer and is a colorless dense sap present inside the nucleus known as nucleoplasm. Nucleoplasm's round shaped nucleus and network of chromatin fibers. Fibers are composed of DNA and protein histone, and these fibers are condensed to form chromosomes during cell division. The right side picture is the nuclear envelope, which is a complex structure consisting of two nuclear membranes and an underlying nuclear lamina and nuclear pore complexes. Two concentric membranes called the inner and outer nuclear membranes. The outer membrane is continuous with the endoplasmic reticulum, so the space between the inner and outer nuclear membrane is directly and connected with the lumen of the endoplasmic reticulum. Now, cytoplasm basically is a thick gel-like semi-transparent fluid that is found in both plant and animal cell. The constituent part of cytoplasm are cytosol, cell organelles, and cytoplasmic incursions. Cytoplasm bounded by the plasma membrane and contains many organelles in eukaryotic cell. Now, cytosol is the aqueous part of the cytoplasm, which is also called a fluid part. Outside of all this contain its own distinctive proteins. It accounts for almost 70% of the total cell volume, and gelatinous substance consisting mainly of cytoskeleton filament molecules, water and salt. Chemically, the cytoplasmic matrix is composed of many chemicals in the form of atoms, ions, and molecules. Okay, the organelles are present in the cytoplasm are mitochondria, endoplasmic reticulum, lysosomes, Golgi apparatus, peroxisomes, and vacuoles. Mitochondria was first discovered in 1850 as a granular structure in the striated muscle and is also called the powerhouse of the cell. And it is also in a filamentous or globular shape. Components of mitochondria are outer membrane, inner membrane, cristae, and matrix. Cristae basically the enfoldings of inner membrane, and matrix is the space enclosed by inner membrane. The membranes are made of phospholipids and proteins. Inner membrane contains ATPase and other enzymes concerned with the synthesis and metabolism of ATP, and the outer membrane is contain large number of integral membrane called porins and is studied with enzymes concerned with biological oxidation. Now, endoplasmic reticulum. Endoplasmic reticulum is a network of tubular and flat vesicular structure in the cytoplasm, and is an space inside the tubules is filled with endoplasmic matrix and an extensive network of closed flattened membrane bounded sac called cristae. Two types of endoplasmic reticulum are there. Number one is a smooth, which is a lack of ribosomes, and rough endoplasmic reticulum is studded with ribosomes. Now, the lysosomes are the irregular structure surrounded by the unit membrane, more acidic than rest of the cytoplasm, and external bacteria as well as worn out cell components are digested in them. The interior is kept acidic by the action of proton pump or hydrogen or ATPase. Lysosomes are cell hydrolysis and they function best at the acidic pH, and it filled with hydrolytic enzymes as well. Now, Golgi bodies is a collection of membrane enclosed sac composed of four or more stuck layers of thin, flat, enclosed vessels lying near the side of the nucleus, and it consists of multiple discrete compartments and also consists of four functionally distinct regions. That is the cis Golgi network, the Golgi stuck, which is divided into the medial and trans subcompartments, and number third is the trans Golgi network as well. Now the peroxisome is a lipid bilayer membrane surrounds which regulates what enter or exit the peroxisomes. Urate oxidase crystalline core structure is similar to that of the lysosomes but with a different